Welcome to Part 2 of our Intracellular Structures Series, prepared for the European Society of Human Reproduction and Embryology Clinical Embryologist Certification. In this session, we turn our attention to a structure so fundamental, so ever-present, that it is easy to take for granted: the plasma membrane. Every cell you will ever work with in the assisted reproductive technology laboratory, every oocyte you assess, every embryo you culture, every sperm cell you evaluate, is defined at its outermost boundary by this remarkable molecular sheet. By the time we finish today, you will appreciate the plasma membrane not as a passive wrapper, but as a dynamic, intelligent interface that governs virtually every clinically significant event in reproductive biology. Let's begin. Consider for a moment just how central the plasma membrane is to everything we do in the assisted reproductive technology laboratory. Every single interaction between a gamete or an embryo and its surrounding environment is mediated through this structure. That is not a rhetorical flourish. It is a literal biological fact. When a sperm cell binds to an oocyte, that binding occurs at the membrane. When the cortical reaction sweeps across the oocyte surface to prevent polyspermy, it is a membrane event. When sperm undergo capacitation in the female reproductive tract or in our culture media, the membrane is where that transformation begins. When a day 5 blastocyst compacts and the cells begin pressing tightly against one another, the membranes of adjacent blastomeres are communicating and adhering. The membrane is not incidental to these processes. It is the stage on which they are performed. And when things go wrong in our procedures, membrane biology is often at the heart of the problem. Cryopreservation, vitrification of oocytes, sperm, and embryos, subjects the plasma membrane to extraordinary osmotic and thermal stress. Whether a cell survives that process depends enormously on how well its membrane can withstand rapid dehydration and rehydration, dramatic temperature shifts, and the presence of cryoprotectant agents that interact directly with lipid components. A membrane that lacks the right lipid composition, or one that has been damaged by oxidative stress, may simply not recover. Beyond cryopreservation, membrane composition directly influences oocyte developmental competence, the capacity of an oocyte to be successfully fertilized, support embryo development, and ultimately result in a healthy live birth. Sperm motility is membrane-dependent, as the flagellar wave requires precise control of ion channels and lipid dynamics along the entire length of the sperm tail. Embryo implantation involves molecular recognition events between the embryonic surface and the endometrium, recognition mediated by membrane-bound glycoproteins and lipids. In short, understanding the plasma membrane is not an abstract academic exercise. It is the foundation upon which your understanding of every assisted reproductive technology procedure must rest. As we move through this session, keep returning to that clinical perspective. Why does this molecule or mechanism matter to the embryos in our incubators right now? Let us take a moment to map the terrain of this session so you know exactly where we are heading. We begin with the architecture of the plasma membrane itself, how the phospholipid bilayer is organized, and what molecular components populate it. From there, we take a brief but important historical detour, examining how our understanding of membrane structure evolved over the 20th century, from the early sandwich model to the fluid mosaic model that remains our conceptual foundation today. We will then spend considerable time on the major membrane phospholipids, 5 key molecular species, each with a distinct structural role and functional personality. We will examine why these molecules assemble into bilayers spontaneously, exploring the elegant thermodynamics of the hydrophobic effect. Cholesterol deserves its own dedicated treatment. Its unique structure, how it positions itself within the bilayer, and how it acts as a master modulator of membrane physical properties across temperature ranges clinically relevant to cryopreservation. We will explore membrane fluidity in depth, what determines it, what regulates it, and critically, what happens to gametes and embryos when it is compromised. And we will close with one of the most functionally significant phenomena in membrane biology: lipid asymmetry. The fact that the 2 leaflets of the bilayer have very different lipid compositions, and that this difference is actively maintained and profoundly important for cell signaling, development, and the safe clearance of apoptotic cells during embryogenesis. There is a great deal of rich material here. Let us begin building our understanding from the ground up. Let us take a moment to map the terrain of this session so you know exactly where we are heading. We begin with the architecture of the plasma membrane itself, how the phospholipid bilayer is organized, and what molecular components populate it. From there, we take a brief but important historical detour, examining how our understanding of membrane structure evolved over the 20th century, from the early sandwich model to the fluid mosaic model that remains our conceptual foundation today. We will then spend considerable time on the major membrane phospholipids, 5 key molecular species, each with a distinct structural role and functional personality. We will examine why these molecules assemble into bilayers spontaneously, exploring the elegant thermodynamics of the hydrophobic effect. Cholesterol deserves its own dedicated treatment. Its unique structure, how it positions itself within the bilayer, and how it acts as a master modulator of membrane physical properties across temperature ranges clinically relevant to cryopreservation. We will explore membrane fluidity in depth, what determines it, what regulates it, and critically, what happens to gametes and embryos when it is compromised. And we will close with one of the most functionally significant phenomena in membrane biology, lipid asymmetry. The fact that the two leaflets of the bilayer have very different lipid compositions, and that this difference is actively maintained and profoundly important for cell signaling, development, and the safe clearance of apoptotic cells during embryogenesis. There is a great deal of rich material here. Let us begin building our understanding from the ground up. Before we dive into the content, let us be explicit about what you should be able to do by the time we finish. These learning objectives are your compass. They tell you not just what to learn, but how deeply to learn it, and in what way that knowledge should be applicable. You should be able to describe the molecular components of the plasma membrane bilayer and explain how they are spatially arranged, not just listing them, but understanding why they sit where they do. You should be able to compare the historical models of membrane structure and articulate clearly why the fluid mosaic model, first proposed by Singer and Nicholson in 1972, remains the conceptual foundation for everything that follows. You should be able to identify each of the 5 major membrane phospholipids, name where in the bilayer each is found, and explain what structural and functional role it plays. Cholesterol warrants its own objective. You should be able to explain precisely how cholesterol regulates membrane fluidity, not in vague terms, but mechanistically, with reference to what it does at high temperatures versus low temperatures and why that matters for cells under thermal stress. You should understand lipid asymmetry, how it is established during membrane biosynthesis, how it is maintained by energy-consuming enzymes, and what the biological consequences are when that asymmetry is lost. And tying everything together, you should be able to connect membrane lipid biology to the clinical outcomes we care about most: oocyte and sperm quality, fertilization success, and cryopreservation survival. Hold these objectives in mind as you work through the session. They are not just examination targets, but the practical toolkit you will carry into the laboratory. The plasma membrane is, at its most fundamental level, a continuous phospholipid bilayer, a sheet of molecules 2 layers thick that completely encloses the cell, separating the intracellular environment from the extracellular world. That separation is the beginning of cellular identity. Without it, there is no chemistry, no homeostasis, no life. But calling it merely a barrier would be like calling a hospital merely a building. What the membrane does with that boundary is what makes it fascinating. The bilayer has a hydrophobic core, a region of densely packed fatty acid chains that are chemically incompatible with water and with the ions and polar molecules dissolved in it. This core acts as a formidable barrier to most water-soluble substances: sodium ions, glucose molecules, amino acids, even water itself to some degree. None of these can simply diffuse through the membrane freely. That selectivity is critical for maintaining the electrochemical gradients and molecular concentrations that cells depend on for every function. But the membrane is emphatically not static. Lipids and many of the proteins embedded within the bilayer diffuse laterally. They move sideways through the plane of the membrane with surprising freedom. This fluidity is not a flaw. It is a design feature that enables the membrane to remodel, to respond, and to recruit the right molecules to the right places at the right times. On the outer surface, carbohydrate chains attached to lipids and proteins form a fuzzy coat called the glycocalyx. This carbohydrate layer serves as a molecular identity badge. It mediates cell-to-cell recognition, protects the membrane from mechanical damage, and plays critical roles in the interactions between sperm and oocyte, and between blastocyst and endometrium during implantation. And woven throughout the bilayer is a diverse population of proteins. These proteins are what transform the membrane from a passive barrier into an active hub of transport, signaling, adhesion, and enzymatic activity. In gametes and embryos specifically, the membrane is the site of fertilization signals, the cortical granule exocytosis reaction, and the cell-to-cell communication that coordinates compaction and differentiation. It is a structure of extraordinary sophistication packed into a sheet just 7 to 10 nanometers thick, less than 1/10,000th the width of a human hair. With that overview in mind, let us look at how scientists came to understand this structure, because the history of membrane biology is itself a lesson in scientific thinking. Every scientific model is a best attempt to explain what we can observe, and the history of how we came to understand membrane structure is a wonderful illustration of how models evolve when new evidence demands it. The story begins in the 1930s and 40s with what became known as the Davison-Danieli sandwich model. Davison and Danieli proposed that the membrane consisted of a central lipid bilayer coated on both surfaces by continuous, uniform layers of protein, much like two slices of bread enclosing a lipid filling. It was an elegant starting point, and it did explain the basic permeability properties of membranes reasonably well. But it came with a serious conceptual problem. It treated membrane proteins as a uniform, static shell wrapped around a passive lipid core. As biochemical and biophysical evidence accumulated through the following decades, that picture became increasingly difficult to defend. Researchers found that membrane proteins varied enormously in type and abundance from cell to cell. They found that some proteins penetrated deeply into or completely through the bilayer. They found that proteins could move, cluster, and redistribute. The sandwich model simply could not accommodate these discoveries. Enter Singer and Nicholson in 1972 with what they named the fluid mosaic model, arguably the most important conceptual advance in membrane biology of the 20th century. Their model described the membrane as a fluid mosaic of amphipathic phospholipids and proteins, all capable of moving laterally within the plane of the bilayer. The lipids formed the flexible, continuous matrix, the fluid part. The proteins were dispersed within that matrix as individual molecules or clusters, like tiles in a mosaic, the mosaic part. Crucially, the model recognized that transmembrane proteins are themselves amphipathic. Their hydrophobic segments span the nonpolar core of the bilayer, while their hydrophilic regions protrude into the aqueous environments on either side. What made this model so powerful was not just that it fit the data available in 1972. It provided a flexible framework that has accommodated decades of subsequent discoveries: lipid rafts, membrane microdomains enriched in cholesterol and sphingomyelin, caveolae, and the specialized junctional complexes that govern cell-to-cell communication. The fluid mosaic model remains our conceptual foundation today, refined and enriched, but not replaced. Now, what exactly is that bilayer made of, and how is it constructed? Let us examine the architecture more closely. The phospholipid bilayer is built from phospholipid molecules arranged in two layers, or leaflets, oriented tail to tail. To understand why this arrangement is so stable and so functional, you need to hold the basic structure of a phospholipid molecule clearly in your mind. Each phospholipid molecule has two distinct regions. The hydrophilic head, composed of a phosphate group and whatever charged or polar group is attached to it, faces outward toward the watery environments on either side of the membrane. The cytosolic leaflet, which is the inner layer, has its head groups facing into the cytoplasm. The non-cytosolic leaflet, the outer layer, has its head groups facing the extracellular fluid. The hydrophobic tails, two fatty acyl chains per molecule, long hydrocarbon strings with no affinity for water whatsoever, face inward toward each other, forming the nonpolar core of the bilayer. This tail-to-tail arrangement shields the hydrophobic chains from the aqueous environments on both sides, achieving a state of minimal energy. What emerges from this arrangement is a remarkably selective barrier. Ions and polar molecules cannot pass through the hydrophobic core without the help of specialized transport proteins. Fat-soluble molecules, by contrast, can dissolve into the core and cross relatively freely. The bilayer is approximately 7 to 10 nanometers thick in total, with the hydrophobic core accounting for roughly 3 to 4 nanometers of that. One of the most important features of the bilayer that we have already touched on but will return to repeatedly is that the two leaflets are not mirror images of each other. Their lipid compositions are distinct. This asymmetry is actively maintained, and it underpins some of the most critical signaling events in reproductive biology. We will explore this in detail later. But first, let us complete our picture of what the bilayer is actually made of, because the lipid and protein components of the membrane are far more diverse than a simple phospholipid scaffold might suggest. When we say the plasma membrane is a mosaic, the diversity of its molecular components is precisely what we mean. The bilayer is not simply phospholipids. It is a carefully regulated assembly of at least 5 categories of molecules, each making a distinct contribution to membrane function. Phospholipids are the major structural lipids. They define the bilayer thickness, determine its curvature, regulate its fluidity, and contribute surface charge. Cholesterol, a steroid lipid, constitutes approximately 20% of plasma membrane lipid by weight in animal cells. Its unique rigid ring structure allows it to modulate membrane fluidity and participate in the formation of specialized ordered microdomains. Glycolipids are lipids with covalently attached sugar chains, and they are found exclusively on the outer leaflet, never on the cytosolic side. Their sugar projections contribute to the glycocalyx, and they participate in cell recognition events, including those critical to sperm-oocyte interaction. Membrane proteins come in 2 broad categories. Integral proteins are embedded within the bilayer, with hydrophobic segments that anchor them in the nonpolar core. These include ion channels, transporters, receptors, and enzymes. Peripheral proteins associate with the membrane surface without penetrating the bilayer, often attaching to the head groups of lipids or to integral proteins. Glycoproteins are proteins with oligosaccharide chains projecting from the extracellular surface. They are major components of the glycocalyx and play pivotal roles in sperm-oocyte binding. The zona pellucida 3 protein, for example, is a glycoprotein that acts as the primary sperm receptor on the oocyte surface. Finally, proteoglycans, core proteins decorated with glycosaminoglycan chains, regulate local signaling environments, contribute to hydration, and provide mechanical protection. Together, these components create a membrane of remarkable functional complexity. Now let us focus our attention on the phospholipid species themselves, because understanding their individual characters is essential for clinical embryology. The 5 major membrane phospholipids are not interchangeable. Each has a distinctive molecular geometry, a preferred location in the bilayer, and a set of functions that makes it irreplaceable. Learning their individual personalities is one of the most important goals of this session. Phosphatidylcholine is the most abundant phospholipid in eukaryotic cell membranes and is found mainly in the outer leaflet. Its cylindrical molecular shape, a head group, and 2 fatty acid tails that together form a roughly rectangular profile makes it ideally suited to forming flat, stable bilayer sheets. It provides the structural backbone of the outer membrane and is the dominant component of the bilayer by abundance. Phosphatidylethanolamine has a smaller head group relative to its 2 tails, giving it a cone-like shape that promotes negative membrane curvature, a slight inward bending of the bilayer surface. This property makes phosphatidylethanolamine critically important wherever the membrane needs to bend sharply, including at vesicle budding sites, during membrane fusion, and during organelle division. It is enriched in the inner leaflet and in mitochondrial membranes. Phosphatidylserine carries a negative charge and is confined to the cytosolic leaflet in healthy, living cells. This confinement is not accidental. It is actively enforced by energy-consuming enzymes, and it is essential for the normal signaling environment of the inner membrane surface. The negative charge on the inner leaflet recruits specific cytosolic signaling proteins that need to dock at the membrane to carry out their functions. And here is the detail that makes phosphatidylserine particularly important in embryology. When a cell undergoes programmed cell death, or apoptosis, phosphatidylserine moves to the outer leaflet, and that translocation is a specific, recognizable signal to the immune system that the cell is ready to be engulfed and cleared. We will return to this in considerable detail when we discuss apoptosis and embryonic tissue homeostasis. Sphingomyelin has a different biochemical backbone from the other phospholipids. It is built on serine rather than glycerol, but its functional role at the outer leaflet is critical. It packs tightly with cholesterol to form lipid rafts, which are ordered signaling microdomains that concentrate specific proteins and lipids together to facilitate efficient signal transduction. Finally, phosphatidylinositol is quantitatively minor but functionally immense. Located on the cytosolic leaflet, its inositol head group can be phosphorylated to generate powerful second messenger molecules. When sperm deliver the enzyme phospholipase C-zeta into the oocyte cytoplasm at fertilization, that enzyme cleaves a phosphorylated form of phosphatidylinositol to generate a molecule called inositol trisphosphate, the trigger for the calcium oscillations that activate the oocyte. Without phosphatidylinositol and its derivatives, fertilization as we know it would not occur. Let us now examine 3 of these phospholipids in closer structural detail. Let us deepen our understanding of the first 3 phospholipid species by thinking carefully about how molecular shape translates into biological function, because in membrane biology, geometry is destiny. Phosphatidylcholine's cylindrical shape is more than a structural convenience. When you have a molecule whose head group area roughly matches its tail area, you get a component that slots neatly into a flat bilayer sheet without introducing any curvature stress. This is exactly what you want for the bulk of the outer leaflet: stability, flatness, and structural integrity. Alterations in the fatty acid composition of phosphatidylcholine, for example, substituting saturated for unsaturated fatty acids, directly affect membrane curvature and the efficiency of vesicle budding. This is clinically relevant when we consider that vesicle trafficking is essential for processes like cortical granule exocytosis during the block to polyspermy. Phosphatidylethanolamines cone shape deserves careful attention because it illustrates an important principle. Membranes do not just need to be flat. In many biological contexts, membranes need to curve dramatically. Vesicle budding, membrane fusion between the sperm and oocyte plasma membranes at fertilization, the division of mitochondria. All of these require controlled, localized bending. Phosphatidylethanolamine, by promoting negative curvature, essentially helps the membrane bend inward. Its enrichment in the inner leaflet and in the highly curved inner mitochondrial membrane is a perfect illustration of structure serving function. Phosphatidylserine requires perhaps the most careful clinical attention of the 3. Its negative charge makes the cytosolic face of the inner leaflet an electronegative surface, and this creates a docking platform for a wide range of cytosolic signaling proteins that carry positively charged domains. These proteins include kinases and other enzymes that coordinate critical signaling cascades within the developing embryo. The strict confinement of phosphatidylserine to the inner leaflet in healthy cells is therefore not just a structural curiosity. It is a signaling environment that must be maintained. Moving forward, let us examine the remaining 2 phospholipid species, sphingomyelin and phosphatidylinositol. Sphingomyelin occupies the outer leaflet of the plasma membrane in particularly high concentrations, and its association with cholesterol is one of the most important relationships in membrane biology. Together, sphingomyelin and cholesterol pack into highly ordered structures called lipid rafts, small, tightly organized microdomains within the otherwise fluid membrane. Think of lipid rafts as specialized platforms. Because they are more ordered and less fluid than the surrounding bilayer, they selectively concentrate certain proteins while excluding others. This selective compartmentalization allows the cell to assemble multi-protein signaling complexes efficiently and to keep different signaling pathways spatially separated so they do not interfere with one another. In sperm, Higher sphingomyelin content in the plasma membrane has been correlated with improved membrane order during the capacitation process. Capacitation involves a carefully orchestrated series of membrane changes that prepare the sperm for the acrosome reaction and fertilization, and the structural integrity provided by sphingomyelin-cholesterol interactions appears to be important for getting that process right. Sphingomyelin also has a metabolic life beyond its structural role. When the membrane is subjected to certain stress signals, sphingomyelin is cleaved by an enzyme called sphingomyelinase to generate ceramide. Ceramide is a bioactive lipid that drives stress responses and contributes to apoptotic signaling. So the same molecule that contributes to structural order in a healthy sperm cell can, when cleaved, initiate cell death, a remarkable duality. Phosphatidylinositol brings us to one of the most functionally extraordinary lipid families in cell biology. Although phosphatidylinositol itself represents only a minor fraction of total membrane lipid, it is the parent molecule for a family of phosphoinositides, phosphorylated derivatives, that serve as pivotal second messengers. Two in particular are central to reproductive biology. Phosphatidylinositol 4,5-bisphosphate, generated by sequential phosphorylation of the inositol head group, is cleaved by phospholipase C-zeta, an enzyme delivered specifically by the sperm, to produce diacylglycerol and inositol 1,4,5-trisphosphate. The latter triggers the release of calcium from the endoplasmic reticulum, producing the calcium oscillations that are the essential trigger for oocyte activation. Without these calcium oscillations, the oocyte remains arrested at metaphase 2 and embryo development cannot begin. This is why calcium ionophore treatment, which artificially triggers calcium release, can rescue oocyte activation failure in assisted reproductive technology cycles where the sperm fails to generate adequate phospholipase C-zeta activity. Understanding this pathway is not academic background knowledge. It is directly relevant to clinical decision-making. With a thorough understanding of our 5 phospholipid species established, let us now examine why these molecules spontaneously organize themselves into bilayers in the first place. One of the most intellectually satisfying aspects of membrane biology is that you do not need a cell to make a membrane. If you simply place phospholipid molecules in water, they will assemble into a bilayer on their own, without any external instruction or energy input. This spontaneous self-assembly is one of the most elegant examples of physical chemistry at work in biology, and understanding it requires grasping the concept of amphipathic molecules and their behavior in water. Phospholipids, glycolipids, and cholesterol are all amphipathic, a term derived from the Greek meaning of both kinds. Each molecule contains a hydrophilic region that is comfortable in water and a hydrophobic region that is emphatically not. When you introduce these molecules into an aqueous environment, a fundamental tension arises. The polar head groups want to interact with water molecules through hydrogen bonds and electrostatic interactions. The fatty acid tails want to be as far away from water as possible. The energetically favored resolution to this tension is for the hydrophobic tails to cluster together, hiding from water, while the head groups orient themselves at the water interface. For phospholipids specifically, the molecular geometry, that roughly cylindrical shape we discussed, strongly favors the formation of bimolecular sheets rather than spherical micelles. In a bilayer, each leaflet presents its head groups to the aqueous phase while the 2 layers of tails pack together in the hydrophobic interior. The result is a flat, stable, selectively permeable sheet. And now here is the additional elegant feature. Bilayers do not like to have exposed edges, because at an edge the hydrophobic tails would be in direct contact with water, thermodynamically unfavorable. So open bilayer sheets spontaneously curve and seal themselves into closed structures, vesicles, organelles, cells. This self-sealing property is the very basis of cellular compartmentalization in all eukaryotes. It also explains why small membrane tears can repair themselves rapidly. The driving force for all of this is what we call the hydrophobic effect, which deserves its own examination. The hydrophobic effect is one of those concepts that seems subtle at first, but turns out to be one of the most powerful organizing principles in biology. Let us think through it carefully, because if you truly understand the thermodynamics, the behavior of membranes under all sorts of conditions, including the extreme conditions of cryopreservation, will make far more intuitive sense. Hydrophilic molecules, sugars, ions, charged amino acids, dissolve readily in water because they carry charged or polar groups that form energetically favorable hydrogen bonds and electrostatic interactions with water molecules. There is a genuine thermodynamic benefit to dissolving these substances in water. Favorable interactions are formed, and the free energy of the system decreases. Hydrophobic molecules, nonpolar, uncharged substances like fatty acid chains, cannot form these favorable interactions with water. But the problem is not just that they fail to form beneficial interactions. The real issue is what happens to the water molecules surrounding them. When a hydrophobic molecule is introduced into water, the water molecules nearby can no longer form their normal network of hydrogen bonds in all directions. Instead, they are forced to reorganize into highly ordered, cage-like structures around the hydrophobic intruder, sometimes called clathrate-like shells. This ordering reduces the entropy of the water significantly, and reducing entropy raises the free energy of the system, which is thermodynamically unfavorable. Here is where the elegance of the hydrophobic effect lies. If multiple hydrophobic molecules cluster together, they reduce the total surface area exposed to water. Fewer water molecules are forced into those ordered cages. Entropy increases. Free energy decreases. The system moves toward a more stable, favorable state. This entropic driving force, the gain in water entropy when hydrophobic surfaces are shielded from water, is the primary thermodynamic engine of bilayer self-assembly. Notice what this means. The membrane does not require continuous energy input to stay assembled. Once formed, the bilayer is thermodynamically stable. It maintains itself. This is a beautiful example of a biological structure that uses the laws of thermodynamics rather than fighting them. Having established why bilayers form and what they are made of, we now turn to one of the most important components embedded within that bilayer: cholesterol. Cholesterol is a molecule that tends to get a bad reputation in popular science. Most people first encounter it in the context of cardiovascular risk and dietary fat. But in membrane biology, cholesterol deserves to be celebrated as one of the most sophisticated and functionally important molecules in the cell. It is the master modulator of membrane physical properties, and its influence on oocytes, sperm, and embryos is profound. Structurally, cholesterol is a steroid lipid built around a rigid four-ring structure called the steroid nucleus. Attached to this rigid core is a small polar hydroxyl group at one end, the hydrophilic portion, and a short hydrophobic hydrocarbon tail at the other. This combination makes cholesterol amphipathic, though in a different way from phospholipids. The molecule is mostly hydrophobic and relatively flat and rigid, quite unlike the flexible, kinked chains of unsaturated phospholipids. In the bilayer, cholesterol orients itself with characteristic precision. The hydroxyl group positions itself near the phospholipid head groups at the membrane surface, forming a hydrogen bond with nearby phospholipid molecules. The rigid steroid rings and the hydrocarbon tail then extend down alongside the fatty acid chains into the hydrophobic core of the bilayer. Crucially, cholesterol occupies the spaces between kinked, unsaturated fatty acid chains, intercalating between them in a way that has significant consequences for how the chains can move. Cholesterol-rich regions of the membrane colocalize with sphingomyelin and glycolipids to form lipid rafts, those ordered signaling microdomains we discussed when examining sphingomyelin. These specialized platforms depend on cholesterol for their existence and their stability. In animal cells, cholesterol constitutes approximately 20% of plasma membrane lipid by weight, a substantial and carefully regulated proportion. Bacteria, notably, lack cholesterol entirely, which is one of the reasons bacterial membranes behave quite differently from animal cell membranes and why drugs that target cholesterol biosynthesis can be selective for eukaryotic cells. Let us now examine exactly how cholesterol regulates fluidity, because that regulation is central to cryobiology. If there is one phrase to anchor your understanding of cholesterol's role in membrane biology, it is this: cholesterol is a fluidity buffer. It does not simply make membranes more fluid or less fluid. Rather, it acts as a stabilizer, dampening the extremes of fluidity across a wide range of temperatures. This buffering function is critical in normal physiology, and it becomes especially relevant when we expose gametes and embryos to the temperature extremes of cryopreservation. Consider what happens to a membrane when temperature rises. At high temperatures, the fatty acid chains of phospholipids gain kinetic energy and move more vigorously. They flex, rotate, and diffuse laterally with greater freedom. The membrane becomes excessively fluid and permeable. Left unchecked, this could cause proteins to lose their functional conformations, disrupt signaling microdomains, and allow ions and molecules to leak across the bilayer inappropriately. Cholesterol counteracts this by inserting its rigid ring structure alongside the fatty acid chains. The rigidity of the steroid nucleus physically restricts the range of motion available to adjacent chains, reducing their lateral diffusion and rotational freedom. The membrane remains functional at temperatures that would otherwise make it dangerously leaky. At low temperatures, the challenge is opposite. Phospholipid chains lose kinetic energy, pack together more tightly, and the membrane risks transitioning from a fluid state into a rigid, gel-like, or even crystalline phase. In a gel phase membrane, protein mobility is severely restricted, membrane fusion is impaired, and the cell cannot carry out the rapid, coordinated membrane events it needs for function. Cholesterol prevents this by inserting between fatty acid chains and disrupting their tight packing, much like a spacer that prevents the chains from crystallizing. Even at low temperatures, cholesterol maintains a degree of membrane flexibility. The clinical relevance of this buffering function is direct and significant. In oocytes, sperm, and embryos, the cholesterol content of the plasma membrane critically influences their ability to survive cryopreservation. Membranes with suboptimal cholesterol content, too little or too much, are more vulnerable to the thermal extremes of vitrification and warming. Furthermore, cholesterol plays a specific role in sperm capacitation. When sperm are exposed to the conditions of the female reproductive tract or to appropriate culture media, cholesterol is extracted from the sperm plasma membrane. And this extraction is actually one of the molecular triggers that initiates the capacitation cascade. The compound cyclodextrin, used experimentally and in some clinical protocols, works precisely by chelating and removing cholesterol from the sperm membrane. Cholesterol's role in reproductive biology, then, extends far beyond structural housekeeping. It is an active participant in the events that lead to fertilization. Let us now examine the broader determinants of membrane fluidity. Membrane fluidity is not a fixed property. It is a regulated parameter that cells adjust continuously in response to changing conditions. There are 4 principal variables that determine the physical state of the membrane, and each one has distinct clinical implications in assisted reproductive technology. The first and most fundamental determinant is fatty acid saturation. The fatty acid chains that make up the hydrophobic tails of phospholipids can be either saturated, meaning every carbon atom in the chain carries the maximum number of hydrogen atoms, and the chain is perfectly straight, or unsaturated, meaning one or more carbon-to-carbon double bonds are present, which introduces a kink in the chain. These kinks are crucial. A straight saturated chain can pack tightly against its neighbors, allowing for close molecular proximity and reduced fluidity. A kinked unsaturated chain, particularly one with a natural cis configuration, cannot pack as tightly. It creates space between neighboring chains, allowing greater freedom of movement and thereby increasing membrane fluidity. Trans fatty acids, which have double bonds in the trans configuration, behave more like saturated fats. Their chains are relatively straight and promote tighter packing and reduced fluidity. Temperature is the second determinant, and its effect is straightforward. Lower temperature reduces the kinetic energy of membrane lipids, causing chains to pack more tightly and the membrane to become more ordered and rigid. Higher temperature does the opposite, increasing molecular disorder and lateral diffusion. What is fascinating is that cells can adapt to chronic temperature changes by adjusting their fatty acid composition, a phenomenon called homeoviscous adaptation. By altering the ratio of saturated to unsaturated fatty acids in response to temperature, Cells maintain relatively constant membrane fluidity despite environmental fluctuations. Cholesterol content is the third determinant, and its temperature-dependent buffering role was our focus in the previous slide. The fourth determinant is oxidative stress, mediated by reactive oxygen species. This is arguably the most clinically important of the four in the context of reproductive aging and gamete quality. Reactive oxygen species, free radicals generated by cellular metabolism and environmental stressors, can attack the double bonds in unsaturated fatty acids, initiating a damaging chain reaction called lipid peroxidation. Peroxidized lipids generate highly reactive breakdown products, including aldehyde molecules that can form crosslinks between membrane lipids and proteins. The result is a bilayer whose normal architecture is disrupted, often becoming more rigid and less functional. This damage is cumulative and, in oocytes that have been arrested in meiosis for years or decades, represents one of the most important mechanisms underlying the decline in oocyte quality with advancing maternal age. With this understanding of fluidity determinants in place, let us examine what happens when fluidity is dysregulated. Membrane fluidity exists within an optimal range, not too fluid, not too rigid. Deviations in either direction have consequences that ripple through molecular, cellular, and ultimately clinical outcomes. Understanding these consequences helps us appreciate why gamete and embryo quality is so sensitive to factors that alter membrane lipid composition. At the level of membrane proteins, excessive rigidity impairs the conformational changes that receptors, ion channels, and transporters must undergo to carry out their functions. A receptor that cannot flex cannot signal. A channel that cannot open cannot conduct ions. Altered membrane order also disrupts the assembly of multi-protein signaling complexes within lipid microdomains, those rafts that depend on precise cholesterol and sphingomyelin packing. When the raft structure is compromised, the spatial organization of signaling cascades breaks down. Signal transduction more broadly is sensitive to membrane order because key signaling lipids, including phosphatidylinositol 4,5-bisphosphate, are concentrated at specific membrane locations, and their availability and activity depend on the physical state of the surrounding bilayer. When that physical state is altered, the spatial logic of signaling is disrupted, with downstream consequences for every pathway that relies on membrane-associated signals. Membrane fusion and division events are particularly vulnerable to fluidity changes. Cortical granule exocytosis, the critical event that establishes the block to polyspermy after fertilization, requires the cortical vesicles to fuse with the plasma membrane rapidly and completely across the entire oocyte surface. This fusion depends on the membrane being sufficiently flexible to undergo the bending and hemifusion intermediates that membrane fusion requires. Too rigid, and the membrane resists bending. Too fluid, and the fusion intermediates become destabilized. The same is true for vesicle trafficking, cytokinesis during embryo cleavage, and the formation of cell-cell junctions during compaction. In the context of reproductive aging, the picture becomes particularly concerning. Cumulative exposure to reactive oxygen species over time drives lipid peroxidation in oocytes, which progressively shifts the membrane toward a more saturated, more rigid lipid profile. As a consequence, calcium release from intracellular stores becomes impaired, reducing the amplitude and number of calcium oscillations at fertilization. Spindle anchoring at the oocyte cortex, which depends on membrane-associated proteins and lipid interactions, becomes less reliable, increasing the risk of chromosome missegregation and aneuploidy. Taken together, these membrane-level changes contribute directly and measurably to the clinical reality that fertilization rates decline, aneuploidy rates rise, and embryo viability decreases as maternal age advances. The membrane is not a bystander in reproductive aging. It is a central participant. Here we arrive at one of the most intellectually compelling aspects of membrane biology, the fact that the two leaflets of the plasma membrane are not mirror images of each other. Their lipid compositions are profoundly different, and that difference is not random or accidental. It is actively generated, continuously maintained by energy-consuming enzymes, and functionally critical in ways that have direct consequences for signaling, apoptosis, and embryonic development. The story begins with lipid biosynthesis. Phospholipids are synthesized primarily on the cytosolic leaflet of the endoplasmic reticulum membrane. They are made facing inward, toward the cytosol. At this stage of their lives, they can move relatively freely between the two leaflets of the endoplasmic reticulum membrane because enzymes called scramblices in the endoplasmic reticulum allow lipids to flip bidirectionally. The endoplasmic reticulum membrane is therefore roughly symmetric. Lipids are distributed fairly evenly between the two leaflets. But as membrane lipids travel via vesicular transport from the endoplasmic reticulum through the Golgi apparatus to the plasma membrane, asymmetry is imposed and then strictly enforced. A family of enzymes called p,4-adenosine triphosphate SE flippases, also known simply as flippases, use the energy of adenosine triphosphate hydrolysis to actively transport phosphatidylserine and phosphatidylethanolamine from the outer leaflet to the cytosolic leaflet. They work against the concentration gradient, maintaining a situation where the inner leaflet is enriched in these two lipids while the outer leaflet is enriched in phosphatidylcholine, sphingomyelin, and glycolipids. The result is a bilayer with an inner leaflet that carries a net negative charge, contributed by the negatively charged head groups of phosphatidylserine and phosphatidylinositol, and an outer leaflet that is relatively neutral and rich in carbohydrate-bearing lipids. This negative inner surface charge is not cosmetic. It creates a docking environment for cytosolic signaling proteins that carry positively charged domains, ensuring that key enzymes and adapters are correctly positioned at the inner membrane face where they need to act. Flippase activity is regulated by adenosine triphosphate availability, calcium concentration, and specific signaling pathways, meaning the cell can adjust the degree of lipid asymmetry in response to physiological cues. When flippases are disrupted, phosphatidylserine appears on the outer leaflet, and as we are about to see, that has profound consequences. Before we close this session, let us consolidate the key concepts we have covered, because the richness of this material means it benefits from deliberate recapitulation. We have established that the plasma membrane is a dynamic fluid mosaic, not a static barrier, but a living, responsive interface built on a phospholipid bilayer. The fluid mosaic model, which remains our conceptual foundation following Singer and Nicholson's landmark 1972 proposal, accounts for lateral mobility of both lipids and proteins, the existence of signaling microdomains, and the dynamic remodeling that underpins every major cellular event in reproductive biology. We have examined the 5 major phospholipid species in detail. Phosphatidylcholine provides structural stability at the outer leaflet. Phosphatidylethanolamine promotes membrane curvature and is essential for fusion events. Phosphatidylserine carries negative charge at the inner leaflet and serves as the apoptotic eat-me signal when externalized. Sphingomyelin partners with cholesterol to form lipid rafts and is metabolized to the signaling lipid ceramide. And phosphatidylinositol, despite its relatively low abundance, generates the phosphoinositides second messengers that are indispensable for fertilization-associated calcium signaling. We have seen that amphipathic lipids self-assemble into sealed bilayers spontaneously, driven by the hydrophobic effect, the thermodynamic gain from increasing water entropy when nonpolar surfaces cluster together. Cholesterol acts as a fluidity buffer, preventing membranes from becoming too rigid at low temperatures and too fluid at high temperatures, with direct relevance to cryopreservation and capacitation. Membrane fluidity is regulated by fatty acid saturation, temperature, cholesterol content, and oxidative stress, all variables that are pertinent to gamete quality assessment and assisted reproductive technology outcomes. Lipid asymmetry is actively maintained by flippases and is essential for the correct signaling environment at the inner membrane surface, for vesicle trafficking, and for the controlled apoptotic clearance that is indispensable for embryonic tissue homeostasis. These are not isolated facts. They form an interconnected understanding of how the membrane works, why it matters clinically, and how its dysfunction contributes to impaired gamete quality and embryo development. This brings us to the end of part 2 of our Intracellular Structures series. You have now built a solid foundation in the structural and lipid biology of the plasma membrane, the molecular architecture that underlies every clinically significant event you will encounter in the assisted reproductive technology laboratory, from oocyte maturation through fertilization, embryo development, and cryopreservation. In part 3, we will extend this foundation by turning our attention to the proteins of the plasma membrane, how integral and peripheral proteins are organized within the bilayer, how they mediate the transport of ions and molecules across what would otherwise be an impermeable barrier, and how their dysfunction connects to the clinical challenges we face in reproductive medicine. The molecular machinery of the membrane is only getting more interesting from here. We will see you in part 3. Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.